All right, so let's kick off this deep dive with something to really make you think.
Sounds intriguing. I'm all ears.
Okay, picture this. You're a scuba diver, exploring a vibrant coral reef. You accidentally brush against a seemingly harmless cone snail, specifically Conicus geographus.
Oh, I know where this is going. That's not a good situation to be in. Those snails pack a serious punch, don't they?
Absolutely. You'd think something so small would be harmless, but within minutes, you could be paralyzed, struggling to breathe. And tragically, many divers have succumbed to this silent assassin of the sea.
It's a stark reminder that it's not always size or brute force that matters.
Precisely. The cone snail's venom is incredibly potent, targeting something truly fundamental to our existence, the way our own nerve cells communicate.
I think that's a perfect segue into our topic today. Neurons, synapses, and signaling. This deep dive is going to take us on a journey into the inner workings of our nervous system.
Exactly. And we have a fantastic chapter packed with information to guide us. Our mission is to distill the essence of how these microscopic marvels work, from their intricate structure to the elegant language of electricity and chemicals they use to keep us functioning.
We'll be exploring how they generate and maintain their electrical charge, almost like tiny living batteries. We'll unravel the mysteries of action potentials, those rapid-fire signals that zip along nerve fibers. And we'll delve into the intricate world of synapses, those microscopic junctions where neurons communicate with each other and with other cells.
Buckle up, everyone. We're about to uncover the secrets of these microscopic powerhouses that drive everything from our reflexes to our thoughts. And the coolest part, we're going to make it clear and engaging, even if you're not a neuroscientist.
Absolutely. This isn't just an academic exercise. Understanding how neurons work is fundamental to understanding how we experience reality. Everything we do, from sensing a gentle breeze on our skin to composing a symphony, originates from these intricate neuronal processes.
It's the language of life at its most basic and most sophisticated levels. So where do we begin?
Well, we need to start with the star of the show, the neuron itself. Picture a cell that's evolved over millennia to specialize in one primary task: receiving and transmitting information. Its structure is a beautiful testament to how form follows function in biology.
All right, so let's break down this specialized structure. We have the cell body, or soma, which acts like the central hub. It houses the nucleus, the mitochondria, and all the essential machinery you'd find in any typical cell.
Right, but what sets neurons apart are these incredible branch-like extensions that radiate outwards from the cell body. These are the dendrites. I always imagine them as a tree's sprawling branches, reaching out to gather signals from countless other neurons. They're like the neuron's sensory antennas, constantly receiving input from its surroundings.
A fantastic analogy.
Yeah, man.
Now, typically extending from the cell body is a single, often much longer projection called the axon. If dendrites are the receivers, the axon is the transmitter, carrying signals away from the cell body to other cells.
And what's mind-boggling is the sheer length some axons can reach. Think of a giraffe. Those nerve cells stretching from its spinal cord all the way down to its hooves can be well over a meter long.
It's like a biological wire, transmitting vital information across incredible distances. And to achieve that rapid communication, they utilize electrical pulses, like a biological telegraph, carrying messages across the body.
Exactly. Now, this axon doesn't just end abruptly. It branches out at its tip into numerous synaptic terminals. Each of these terminals forms a synapse, that crucial junction where the neuron's signal is relayed to another cell, almost like a microscopic handshake between cells.
So we have the neuron sending out its axon, which branches into these terminals, forming synapses with other cells. But how does the signal actually cross that gap between the neuron and its target?
That's where things get even more fascinating. At most synapses, the communication isn't directly electrical, but rather chemical. This is where neurotransmitters come into play. These are the chemical messengers that bridge the synaptic cleft, the tiny gap between the presynaptic neuron and the postsynaptic cell.
It's like passing tiny notes across a gap to relay information. And remember our cone snail from the beginning. Its venom cleverly disrupts both the electrical signals traveling along the axon and the chemical signaling at the synapse, essentially shutting down communication within the nervous system.
A precise and deadly attack on the very essence of how the nervous system functions. Now, let's take a step back and consider how a neuron processes information. It's a beautifully elegant three-stage process: sensory input, integration, and motor output.
Think of the cone snail again. Its siphon acts as a highly sensitive chemical detector, gathering information from the surrounding water. That's the sensory input.
Then, within its rudimentary nervous system, a network of neurons analyzes this information, deciding whether it's a potential threat or prey. That's the integration stage.
And finally, if it detects a tasty morsel, it sends out commands to its harpoon mechanism to capture the prey. That's the motor output.
In more complex creatures like ourselves, we see a division of labor with specialized types of neurons handling each stage. Sensory neurons act like the body's reporters, relaying information about the external world and our internal state to the central nervous system.
And then we have the inner neurons, those densely packed processing units primarily found in the brain and spinal cord. They analyze the incoming information, make decisions, and orchestrate appropriate responses.
And finally, motor neurons act as the messengers, carrying instructions from the CNS to the muscles and glands, the effectors that carry out the body's actions.
Now, here's something I find truly fascinating. The electrical signals within a neuron are remarkably uniform, regardless of whether it's a sensory neuron, an inner neuron, or a motor neuron. It's the connections they make, the specific pathways they form, and the strength of those connections that determine the information being conveyed.
It's like a language where the alphabet is the same for everyone, but the meaning comes from how we arrange the letters into words and sentences. The intricate web of connections between neurons creates the richness and complexity of our nervous system.
Absolutely, and this functional specialization is beautifully reflected in the sheer diversity of neuron shapes and sizes. Some, like bipolar neurons, are relatively simple. Others, like the Purkinje cells in the cerebellum, have incredibly elaborate dendritic trees, like a neuron with thousands of tiny fingers reaching out to receive information.
Each neuron's shape reflects its unique role within the nervous system, and these bundles of axons, those biological wires we talked about, are what we collectively call nerves, forming the intricate communication highways throughout the body.
Right. And then we have the major organizational divisions within our nervous system. The central nervous system, or CNS, and the peripheral nervous system, or PNS.
The CNS, consisting of the brain and spinal cord in vertebrates, is the central command center. The PNS is the vast network of nerves extending outward from the CNS, carrying sensory information in and motor commands out, like the communication lines connecting the brain and spinal cord to the rest of the body.
But we can't forget about the unsung heroes of the nervous system, the glial cells, or glia.
The name itself, from the Greek for glue, hints at their traditional role as structural support cells, holding neurons in place. But as research has progressed, we've come to appreciate that they do so much more than just provide scaffolding.
If you could peek inside the brain with a powerful microscope, you'd see a vibrant, bustling scene filled with glial cells alongside the neurons. Imagine a bustling city, but at a microscopic scale. The glial cells are like the tireless support staff, outnumbering the neurons, ensuring everything runs smoothly.
They nourish neurons, insulate axons, regulate the chemical environment surrounding neurons, and play a crucial role in immune defense within the nervous system. They're essential for proper nervous system development and function.
All right, so we've laid the groundwork, understanding the basic structure of neurons and their supporting cast. Now let's delve into the heart of how they communicate, the electrical signaling. And at the core of this is the concept of membrane potential.
Imagine the plasma membrane of a neuron not just as a physical barrier, but as a dynamic electrical boundary. There's a difference in electrical charge or voltage across this membrane. It's like a tiny biological battery holding a potential difference between the inside and outside of the cell.
And this voltage arises from the unequal distribution of ions, those charged atoms, across the membrane. In a neuron that's not actively sending a signal, we call this the resting state, the inside of the cell is typically more negative compared to the outside.
This voltage across the membrane in the resting state is what we call the resting potential, typically falling within the range of -60 to -80 millivolts. Though it might seem minuscule, this resting potential is absolutely crucial for a neuron's ability to generate and transmit signals.
Now, this unequal distribution of ions that creates the resting potential is primarily due to two key players, potassium ions, or K plus man, and sodium ions, or name plus mean.
If we peek at the concentrations of these ions inside versus outside the neuron, we see a striking difference. Potassium is much more concentrated inside the cell, while sodium is predominantly found outside.
And this difference in concentration is crucial. If there's a higher concentration of an ion on one side of a membrane, it naturally wants to move to the side with the lower concentration to even things out. Like diffusion, things tend to move from where they're crowded to where there's more space.
But the cell membrane isn't just a simple sieve. It's selectively permeable, meaning it carefully controls what can pass through. This is where ion channels come into play. These are protein channels embedded in the membrane that act like selective gates, allowing only specific ions to pass.
And so we have these concentration gradients pushing ions to move, and we have these ion channels that regulate their movement across the membrane. And there's another force at play here, right?
Exactly. As ions move across the membrane, they carry their electrical charge with them. So if positive ions move out of the cell, the inside becomes more negative. And this buildup of charge creates an electrical force that can oppose the movement of ions driven by their concentration gradients.
It's like a tug-of-war between the chemical force from the concentration gradient and the electrical force from the charge difference.
Precisely. And for each type of ion, there's particular membrane potential at which these two forces balance each other out. We call this the equilibrium potential for that ion. It's the voltage at which there's no net movement of that ion across the membrane.
So for potassium, the equilibrium potential is around 90 millivolts. This means that if the inside of the cell were 90 millivolts more negative than the outside, there would be no net flow of potassium ions in or out.
Now for sodium, the equilibrium potential is about plus 62 millivolts. So sodium would be at equilibrium if the inside of the cell were 62 millivolts more positive than the outside.
But remember, the actual resting potential of a neuron is somewhere between -60 and -80 millivolts. Why isn't it exactly at the equilibrium potential for either potassium or sodium?
That's because the membrane is much more permeable to potassium ions than to sodium ions at rest. There are many more open potassium channels, allowing potassium to leak out of the cell more readily. This outward flow of positive potassium ions makes the inside of the cell more negative, driving the membrane potential closer to the equilibrium potential for potassium.
So it's like potassium has a stronger influence on the resting potential because it's freer to move across the membrane.
Exactly, but the membrane isn't completely impermeable to sodium either. There are still a few open sodium channels, allowing a trickle of positive sodium ions to enter the cell. This slight inward flow of sodium counteracts some of the negative charge buildup, making the resting potential slightly less negative than the equilibrium potential for potassium.
Now, even though there's this constant movement of ions potassium leaking out, sodium trickling in the overall concentrations of these ions, inside and outside the cell, remain remarkably stable. How is that possible?
It's a delicate balancing act. The key player here is the sodium-potassium pump. This incredible molecular machine embedded in the membrane actively pumps sodium ions out of the cell and potassium ions back in, working against their concentration gradients.
It's like a tiny pump constantly working to maintain the unequal distribution of ions that creates the resting potential. And this pump requires energy to do its job, using ATP, the cell's energy currency, to power its actions.
Exactly. While the pump itself contributes a small part directly to the resting potential, its primary role is to establish and maintain those crucial ion concentration gradients that make the whole system work. Now, we've talked about these always open leak channels, but there's another important category of ion channels: gated ion channels.
These are like doors that can open or close in response to specific signals. For example, some channels are voltage gated, meaning they open or close depending on changes in the membrane potential itself.
Others might be chemically gated, meaning they respond to the binding of specific molecules, like neurotransmitters. And when these gated channels open or close, they alter the flow of ions across the membrane, changing the membrane potential in localized areas.
So if the inside of the neuron becomes even more negative than the resting potential, maybe because more potassium channels open, we call that hyperpolarization.
Right. It's like making the inside of the cell even more different from the outside in terms of charge. And conversely, if the inside becomes less negative, perhaps due to the opening of sodium channels, we call that depolarization.
It's like bringing the charge inside and outside the cell closer together. And these localized changes in membrane potential hyperpolarizations and depolarizations are called graded potentials.
And they're called graded because their strength varies depending on the strength of the stimulus. A stronger stimulus will cause a larger change in membrane potential. Think of it like ripples in a pond. The bigger the splash, the bigger the ripples.
But these graded potentials tend to fade out as they spread away from their point of origin. They're like whispers that diminish with distance.
But if a depolarizing graded potential is strong enough and happens at a particular location on the neuron, the axon hillock, that cone-shaped region where the axon arises from the cell body, it triggers something much more dramatic: the action potential.
And this is the main event: the rapid, long-distance electrical signal that neurons use to transmit information over long distances.
Precisely. The action potential is like a shout that can travel along the entire length of the axon without fading away.
Now, there's a critical level of depolarization, a threshold, that needs to be reached at the axon hillock to initiate an action potential.
And once that threshold is crossed, it triggers an all-or-none response. This means that the action potential always fires with the same magnitude, regardless of how strong the stimulus was beyond that threshold. It's like flipping a light switch. It's either on or off. There's no in-between.
So what's happening at the molecular level to generate this all-or-none action potential?
Well, it's a beautifully orchestrated dance of voltage-gated ion channels, those doors we talked about, that open and close in response to changes in membrane potential.
Let's break it down step by step. Imagine the neuron at rest, with its resting potential established. The majority of the voltage-gated sodium channels are closed, but ready to spring into action. Only the potassium-leak channels are open, maintaining that resting potential.
Now, along comes a depolarizing stimulus, a graded potential that nudges the membrane potential at the axon hillock toward the threshold. If it's strong enough to reach that threshold, a cascade of events is triggered.
First, a bunch of voltage-gated sodium channels quickly open. Sodium ions, driven by both their concentration gradient and the negative charge inside the cell, rush into the neuron. This massive influx of positive charge causes a rapid depolarization, sending the membrane potential soaring upwards.
It's like a dam suddenly bursting, with a torrent of sodium ions flooding into the cell. But this can't continue indefinitely.
Right, and that's where the falling phase of the action potential kicks in. Two things happen to bring the membrane potential back down. First, the voltage-gated sodium channels have a built-in timer, and after a brief period, they inactivate, shutting off the flow of sodium ions. It's like the dam slamming shut.
And second, with a slight delay, voltage-gated potassium channels finally open wide. Potassium ions, now driven by their concentration gradient and the positive charge inside the cell, flow out of the neuron. This outflow of positive charge repolarizes the membrane, bringing the potential back down towards the resting state.
It's like opening a release valve, allowing the excess positive charge to flow out, restoring balance. But there's a little blip at the end, right? A brief overshoot where the membrane potential becomes even more negative than the resting potential.
Exactly. That's called the undershoot, or hyperpolarization. It happens because those potassium channels stay open a bit longer than needed, allowing a little extra potassium to flow out before they close.
And then finally, the sodium-potassium pumps kick in, restoring the original ion concentrations and bringing the membrane potential back to its resting state, ready for the next action potential.
And that inactivation of the sodium channels is crucial because it prevents the action potential from traveling backwards along the axon. It ensures the signal flows in one direction only, from the axon hillock towards the synaptic terminals.
It's like a one-way street for the electrical signal. Now, the action potential doesn't just sit there. It propagates, traveling down the axon like a wave. But how does it actually move?
Well, as the action potential occurs at one point on the axon, the influx of sodium ions creates a local electrical current that spreads to the adjacent section of the membrane. If this depolarization reaches the threshold in that neighboring region, it triggers a new action potential there, and so on, down the entire length of the axon.
So it's like a chain reaction.
Yeah.
With each action potential triggering the next one down the line.
Precisely. And because the region behind the traveling action potential is still in its refractory period, remember, those sodium channels are still inactivated. The signal can only travel forward, not backward.
It's like a line of dominoes falling one after another in only one direction. Now, we know that the intensity of a stimulus, like a bright light or a loud sound, isn't encoded in the size of the action potential itself, since it's an all or none event. So how does the nervous system convey information about the strength of a stimulus?
It's all about frequency. A stronger stimulus will trigger a higher frequency of action potentials. It's like sending more signals per second to indicate a more intense input.
So if you touch a hot stove, the sensory neurons in your hand will fire off action potentials at a much faster rate than if you touch something lukewarm.
Precisely. The brain interprets this higher frequency of signals as a more intense sensation. Now, we've been talking about how action potentials travel along individual axons, but axons can be quite long. How do they transmit signals efficiently over long distances without losing strength?
That's where myelination comes in, one of nature's ingenious solutions for optimizing signal transmission.
Imagine the axon as an electrical wire. Myelin is like an insulating sheath wrapped around that wire, preventing the electrical signal from leaking out.
In the CNS, this myelin sheath is formed by specialized glial cells called oligodendrocytes. In the PNS, it's formed by Schwann cells.
And myelin isn't continuous along the entire length of the axon. There are these periodic gaps called nodes of Ranvier. These nodes are the only points where the axon membrane is directly exposed to the extracellular fluid.
And critically, these nodes are packed with voltage-gated sodium channels. So instead of having to regenerate the action potential at every point along the axon, the signal can jump rapidly from one node to the next.
This process is called saltatory conduction, from the Latin word saltare, meaning to leap. It's a remarkably efficient way to speed up signal transmission, as the action potential doesn't have to slowly regenerate along the entire length of the axon.
It's like taking a high-speed train that only stops at major stations, instead of a local train that stops at every little town along the way.
Precisely. And this myelination is crucial for proper nervous system function. In diseases like multiple sclerosis, where the myelin sheath is damaged, nerve signal transmission is disrupted, leading to a variety of neurological symptoms.
It highlights just how important this insulation is for the rapid and reliable communication within the nervous system.
Now, imagine that action potential, having raced down the axon, finally reaches its destination, the synaptic terminal. What happens next?
That's where the magic of synaptic transmission unfolds, the process of communication between neurons. We've briefly touched on synapses, those microscopic junctions where neurons connect with each other or with other cells.
And as we mentioned earlier, most synapses in our nervous system are chemical synapses, relying on those chemical messengers we call neurotransmitters.
So the action potential arrives at the synaptic terminal, and this triggers a cascade of events that leads to the release of neurotransmitters into the synaptic cleft.
Here's how it works. The arrival of the action potential depolarizes the membrane at the synaptic terminal. This depolarization opens voltage-gated calcium channels, and calcium ions rush into the terminal.
And this influx of calcium is the critical trigger for neurotransmitter release. It causes tiny membrane-bound sacs called synaptic vesicles, filled with neurotransmitters, to fuse with the presynaptic membrane and release their contents into the synaptic cleft.
Imagine these synaptic vesicles like tiny cargo ships docking and unloading their precious neurotransmitter cargo into the space between the two neurons.
Then these neurotransmitter molecules diffuse across the synaptic cleft and bind to specific receptor proteins on the postsynaptic membrane, the membrane of the receiving cell.
It's like a lock-and-key mechanism. Each neurotransmitter has a unique shape that fits only into its corresponding receptor. And this binding triggers a response in the postsynaptic cell.
Now, these postsynaptic responses can be either excitatory or inhibitory.
Right, exactly. An excitatory postsynaptic potential, or EPSP, depolarizes the postsynaptic membrane, bringing it closer to the threshold for firing its own action potential. It's like giving the postsynaptic neuron a little nudge towards generating its own signal.
And an inhibitory postsynaptic potential, or IPSP, does the opposite. It hyperpolarizes the membrane, moving it further away from the threshold. It's like putting on the brakes, making it less likely for the postsynaptic neuron to fire.
And these postsynaptic potentials are graded potentials, meaning their strength varies depending on the amount of neurotransmitter released and the number of receptors activated.
Now, the neurotransmitter signal can't just linger in the synaptic cleft forever. There needs to be a way to turn it off, allowing for precise control of synaptic transmission.
Right. There are several mechanisms for clearing the neurotransmitter from the synaptic cleft. One is enzymatic degradation, where specific enzymes break down the neurotransmitter into inactive components.
For example, the enzyme acetylcholinesterase rapidly breaks down the neurotransmitter acetylcholine, ensuring the signal is quickly terminated.
Another mechanism is reuptake, where the presynaptic neuron, or sometimes nearby glial cells, actively transport the neurotransmitter back into the cell. It's like recycling those precious messenger molecules.
And this efficient clearance is crucial. I recall reading about nerve gases that block the breakdown of acetylcholine, leading to a buildup of the neurotransmitter at neuromuscular junctions, causing prolonged muscle contraction and paralysis. It's a terrifying illustration of how disrupting this delicate balance can have devastating consequences.
Absolutely. Now, we've primarily focused on ionotropic receptors, those receptors that are directly coupled to ion channels. But there's another important class, metabotropic receptors. These receptors work indirectly, triggering a cascade of intracellular signaling events that ultimately lead to changes in the postsynaptic neuron.
It's like a chain reaction inside the cell. with the neurotransmitter binding setting off a series of events that can lead to a variety of responses, including the opening or closing of ion channels, changes in gene expression, or the activation of other enzymes.
And these metabotropic responses tend to be slower and longer lasting than the direct responses mediated by inotropic receptors.
It's like the difference between flipping a light switch on and off and slowly dimming the lights.
Now, the nervous system uses a remarkable diversity of neurotransmitters, each with its own unique set of receptors and effects. This incredible variety allows for the nuanced and complex communication that underlies everything our nervous system does.
For example, acetylcholine is crucial for muscle contraction at the neuromuscular junction, but it also plays important roles in learning and memory in the brain.
Glutamate is the primary excitatory neurotransmitter in the brain, involved in everything from sensory perception to learning and memory.
And GABA is the main inhibitory neurotransmitter, helping to keep the brain's activity in check and prevent overexcitation.
And there are many others, each with its own fascinating story. For example, dopamine is involved in reward and motivation, and serotonin plays a key role in mood regulation.
Now, a single neuron within the brain can receive input from thousands of other neurons, some excitatory and some inhibitory. So how does it integrate all this information and decide whether or not to fire its own action potential?
That's where the concept of summation comes in. It's the process of adding up all the incoming excitatory and inhibitory signals to determine the overall effect on the postsynaptic neuron.
Think of it like a little calculator inside the neuron, constantly adding and subtracting the incoming signals.
There are two main types of summation: temporal summation and spatial summation. Temporal summation occurs when multiple signals from the same presynaptic neuron arrive in rapid succession, their effects adding up over time.
It's like repeatedly tapping on a door. Each tap might not be enough to open it, but many taps in a row will eventually do the trick.
Spatial summation, on the other hand, occurs when signals from multiple different presynaptic neurons arrive simultaneously at different locations on the postsynaptic neuron.
It's like having a group of people pushing on a door from different angles. Their combined effort might be enough to open it, even if no single person could do it alone.
The axon hillock, that critical trigger zone for action potentials, acts like a miniature integration center, constantly summing up all the incoming excitatory and inhibitory signals. If the net effect is sufficient to depolarize the membrane to the threshold, an action potential is generated and sent racing down the axon.
It's a remarkable system, allowing neurons to make sophisticated decisions based on a complex interplay of excitatory and inhibitory inputs.
And this intricate interplay of neuronal signaling underlies everything our nervous system does, from the simplest reflexes to our most complex thoughts and emotions.
So, to wrap things up for you, dear listener, we've embarked on a fascinating journey into the inner workings of the nervous system. We've explored the intricate structure of neurons, those specialized cells designed for communication.
We've delved into the world of electrical signaling, from the resting potential to the all-or-none action potential that zips along axons, and how myelination speeds up transmission over long distances.
And we've uncovered the secrets of synaptic transmission, where neurons communicate with each other using a diverse array of chemical messengers.
Understanding these fundamental processes is not just a scientific curiosity. It's the key to understanding how we perceive the world, think, feel, and act.
From the simplest reflex to the most profound creative thought, it all stems from these intricate interactions between neurons.
And as we've seen with the cone snail's venom, disrupting this delicate balance can have profound consequences.
So here's a final thought for you to ponder. Given the incredible complexity and diversity of neurotransmitters and their receptors, and the profound influence they exert on every aspect of our being, what untapped potential might lie in developing new therapies and interventions that target specific neurotransmitter systems?
Imagine the possibilities for treating neurological disorders like Parkinson's and Alzheimer's, for developing more effective antidepressants and anti-anxiety medications, or even for enhancing cognitive function and creativity.
It's a fascinating and rapidly evolving field, and we've only just begun to scratch the surface of what we can learn about the intricate workings of our nervous system. We encourage you to continue exploring this captivating world, diving deeper into the mysteries of the brain and unlocking the secrets of who we are.
It's an exciting journey of discovery, and we hope this deep dive has sparked your curiosity and inspired you to learn more.